All right, so in this video lecture, we're going to look at your erythrocytes or red blood cells. We're going to look at the structure and function of them, talk about the hemoglobin that's contained inside of them, and then look at what nutritional requirements are needed to make red blood cells and hemoglobin, how are numbers of erythrocytes maintained or regulated, and then what is the life cycle of a erythrocyte. So let's start with some just general characteristics about red blood cells. First of all, during their maturation or the development, they've lost all the internal organelles or the cytoplasmic organelles, which means they can't maintain themselves. If they become damaged, that means they don't live very long, only about 120 days. They're also disc-shaped. That is, you can see this biconcave shape on each side. That makes them more flexible and stackable, so they're like pancakes, so they can move through small capillaries more readily. Their function is for gas transport, so that biconcave actually increases the surface area of the red blood cells so they can get maximum movement of oxygen in and out of those red blood cells. Now, 33% of the cytoplasm in a red blood cell is hemoglobin, and again, that's for oxygen delivery and carbon dioxide transport. And also inside that red blood cell is going to be carbonic anhydrase. If you remember, we talked about this as the enzyme that causes the combination of carbon dioxide and water to convert it into carbonic acid. And so this is one of those, that equation that's going to be important as we look at gas transport and pH balance. Now here's just a picture of some red blood cells on the tip of the needle so you can get a perspective on how teeny tiny they actually are. If we look at the hemoglobin structure, hemoglobin is composed of 4 protein chains called globins. This is a good example of a quaternary structure in a protein. So here we've got 2 alpha chains here in the lighter color and 2 beta chains in the darker color. Okay, so those are the 4 chains that make up, or the 4 protein chains or globins that make up hemoglobin. In the middle of each protein is a molecule called heme, or a heme group. If I look at that molecular structure of a heme group, it looks like this, and you can see buried in the very middle of it is an iron molecule. That iron is where the oxygen binds to it. So if I've got 4 hemes, I've got 4 irons in the middle. I can bind 1 oxygen to each iron, so that means hemoglobin can carry, 1 molecule of hemoglobin can carry 4 oxygen molecules is all they can carry. Now when oxygen is carried on the hemoglobin, we call it oxyhemoglobin. When there is no oxygen bound to the hemoglobin, we call it deoxyhemoglobin. Now, carbon dioxide can also be carried by the hemoglobin, but not on the iron or on the heme. Carbon dioxide binds to the amino acid of the globin, and so that happens. Then we get carbaminohemoglobin. Now, the red blood cell count and hemoglobin concentrations give us an idea of the amount of oxygen the blood can carry. So it's important that in a patient you measure how much hemoglobin or how much oxygen they could possibly carry. So we'll look at a couple ways we can measure that indirectly by looking at characteristics of the red blood cells. So first, the hematocrit. We've already seen hematocrit in the previous lecture. Hematocrit is basically the proportion of the blood that's composed of cells. And so for men, roughly 42 to 50%, women, not quite so much, 37 to 48. We can also look at hemoglobin concentration. That is, we can measure how much hemoglobin is in those red blood cells. And so again, men slightly higher than women. And finally, we can look at red blood cell count. That is, literally count how many they are per microliter. And again, men higher than women. Now, why are the values lower in women? Well, 1, men have androgens like testosterone, which stimulate red blood cell production, so they end up with more red blood cells and higher hemoglobin content, that type of thing, which they need because they have a higher metabolism than we do, and because they have higher muscle mass than we do. So they have to have all that hemoglobin to supply those muscles with oxygen. Women have also our periods, our menstrual cycles, prevent us from having any huge amounts since we become anemic every month when we have our periods. Now, erythropoiesis is the production of red blood cells. It's a multi-step process, and you can see here the stages that undergoes. Not that I want you to learn the names of those guys, but what we're looking at is some general trends instead. Now, erythropoiesis happens very rapidly in that we make 2.5 million red blood cells per second, so that's a lot. It takes a while, though. It takes about 15 days to go through all those processes. But in that process, you can see, first of all, a reduction in cell size. So as we go left to right, the cells gain smaller. Also, you can see that we've lost the nucleus. So by the time we get to the stage called the reticulocyte, the nucleus is gone, or you only see little fragments or leftover parts of the nucleus. Now those reticulocytes will actually go into circulation, and you can count those as well when you do a blood cell count, and that will give you an idea of the rate of red blood cell formation. Now, to maintain erythropoiesis is a classic negative feedback kind of mechanism. It's tied in with oxygen levels. So anytime you have low oxygen levels, we're going to trigger the production of more red blood cells to deal with those low oxygen. So up here you can see where hypoxemia means inadequate oxygen transport. So that's going to be detected by the liver and the kidney. Both of them, but primarily the kidney, is going to secrete erythropoietin. This is a hormone that stimulates the red bone marrow to increase erythropoiesis. So now we make more red blood cells. Our red blood cell count goes up. Because we have more red blood cells, we can transport more oxygen. So oxygen level in the blood goes up, and now we're not hypoxemic anymore. Now there's a lot of things, though, that could stimulate erythropoiesis, and a lot of different reasons you may have low oxygen levels. Some are normal, some are not. One is basically you live in low levels of oxygen in the atmosphere, such as maybe you live in Denver where the altitude is much higher, so there's not as much oxygen, so you need more red blood cells to be able to carry the oxygen that is available. Also, increase exercise. If you start exercising more, you increase muscle mass, and that puts a bigger oxygen demand on there, so you need more red blood cells to provide that oxygen. Hemorrhaging obviously isn't the good side of this. Hemorrhaging may happen. You bleed out, you lose a large volume of blood, hence then you don't have as much blood to transport oxygen, so you're going to trigger more erythropoiesis with that. And then could be low levels of hemoglobin in the red blood cell, maybe an iron deficiency or lack of intrinsic factor to get B12, or all kinds of other possibilities that are tied in with what we need to make hemoglobin. Now, there's always going to be a small amount of EPO or erythropoietin in circulation to help sustain red blood cell production. Now, nutritional needs for erythropoiesis, of course, iron is probably the key component to making hemoglobin. We lose iron daily in our feces, in the urine, and bleeding. Men have a smaller loss than women do because, of course, women were menstruating, so we lose more iron on average per day. Iron, though, is not very well absorbed through the digestive tract, so we have to consume like 5 times as much iron as what we actually lose, since we're not going to absorb all that iron we eat. Iron comes in 2 forms, either ferric or ferrous, and then we'll just convert one to the other in the stomach, so it's no big deal which one you get it from. And then, of course, the liver can store excess iron and use it for later. Some other things, though, we need besides iron is B12 and folic acid. This is used for DNA synthesis, so we need these guys, plus some vitamin C and copper to synthesize red blood cells, or frankly, any of the other cells. So we need to make white blood cells or platelets. We need the B12 and folic acid and C and copper as well. Now, if we look at the way we transport and absorb the iron, what I want you to notice, is particularly as I go through this slide, how much iron is bound to something in every step along the way. So we bring in iron in our diet here, And then, as I mentioned, the stomach's just going to convert one to the other. And then that iron is going to be bound to a protein called gastroferritin. So gastro knows stomach. So it's going to, this molecule, gastroferritin, binds up the iron. Now that iron then, it moves into the small intestine with the gastroferritin. There the gastroferritin lets go of it, so the iron can be absorbed into our blood. But as soon as it gets in the blood, it's bound up by another protein called transferrin. Now transferrin keeps the iron bound up in our blood. As we go, it can take it directly to cells if the cells need it for some reason, or it's going to go to the liver and we can store it there in the liver. And the way we store in the liver is another protein called apoferritin binds with the iron and forms ferritin. So again, apoferritin is the protein by itself. Ferritin is the protein plus the iron in combinations, and that's the way we're going to store it in our liver. Now, any transferrin that carries iron along, it could take it to the marrow, and that's where it could be made into more hemoglobin or even myoglobin, which is the way we store oxygen in our muscles. Now, the life cycle of a erythrocyte looks something like this. We've already gone through this side on the left because this talks about red blood cell production when we have low oxygen levels. So this is that negative feedback. That's what the NFB is there. Negative feedback with low oxygen means more erythropoietin, which means more erythropoiesis, which means more red blood cells produced. And so now we can carry more oxygen, so our oxygen levels go back up. Now, once those red blood cells are produced, then they're going to go in circulation for about 120 days. Towards the end of their life, they start getting worn out. They've been bumping up against walls of blood vessels and getting kind of beaten up a little bit, so their membranes aren't very strong. So when these old blood cells, red blood cells particularly, go into the spleen or liver and they have to move through tiny little capillaries, they get roughed up even more and they could rupture. So there's macrophages there to kind of pick up these old crappy red blood cells and eliminate them from circulation. It's a normal process. We want to get rid of these old guys, and so those macrophages are engulf them. And now macrophages, are a type of white blood cell. Now the hemoglobin inside that red blood cell, though, we want to process it and recycle a portion of it because we might as well use it. It's not like the hemoglobin's gone bad, it's just the red blood cells worn out. So let's take portions of the hemoglobin that we can and recycle it. So if you remember, the hemoglobin is made up of globin, those protein molecules. Well, let's just break up those proteins into their amino acids, and we'll recycle the amino acids back into circulation and use them for either making more hemoglobin or for making something else. Then the heme part of the hemoglobin, now this, remember, has iron in the middle of it, so I can pull the iron out of it, take the iron, store it in the liver, or put it back in circulation so it can be used to make more erythrocytes over here in the bone marrow. So I'm going to recycle the iron as well. The rest of the heme is going to be converted into bilirubin. If you remember from digestive system, I mentioned bilirubin before, that bilirubin ends up part of the bile. So it's excreted into our intestines with the bile, and then we can deposit it or excrete it out of our bodies. So the only part of the hemoglobin we can't recycle is that bilirubin, which is the heme minus the iron. Both the globin and the iron can get recycled and used again. Our deficiency in red blood cells or hemoglobin is going to be anemia. So anemia, you see low oxygen levels, but there are a lot of different possible causes. Some of these are related to erythropoiesis or hemoglobin synthesis. One of those would be iron deficiency anemia. This is the most common form of anemia, where you're simply not getting enough iron in your diet, and therefore you can't make hemoglobin, and if you can't make hemoglobin, then you're going to become anemic. There's also pernicious anemia. This is an autoimmune disease that results from lack of intrinsic factor. Remember, intrinsic factor is made by your stomach, but in pernicious anemia, your immune system attacks the cells that make the intrinsic factor, and so therefore they can't make it, and you can't absorb vitamin B12. If I can't get vitamin B12, I can't make red blood cells. As a matter of fact, I can't make any kind of formed element, the white blood cells, platelets, or red blood cells, and so I have a shortage of all of them. That's different than B12 deficiency anemia. B12 deficiency anemia says you simply aren't getting enough B12 in your diet. Now, renal anemia is another form of anemia where the kidneys are failing or damaged for some reason, and so you're not making enough erythropoietin, and without the erythropoietin, we're not stimulating erythropoiesis. Aplastic anemia is another one. This is usually from destruction or inhibition of the red marrow by certain drugs or chemicals or radiation and viruses, or aplastic anemia can result from simply, they don't know. It all of a sudden just shows up and you just don't know what the actual cause of it is. But it can result in all the formed elements, again, the white blood cells, red blood cells, and platelets being reduced, which means then you are deficient in white blood cells, so you can have immunological problems. You're going to be deficient in blood clotting, platelets, so blood clotting is going to be impaired, and of course, since you're short on red blood cells, you have anemia. Another type of anemia is hemorrhagic anemia, which basically means you're bleeding. So you lose a lot of blood, you're going to be low on oxygen levels from that, and not be able to carry oxygen or transport if you don't have enough blood. And then hemolytic anemia is from the destruction of red blood cells, or the over-destruction of red blood cells, that can lead to then hemolytic anemias. And we can see that with sickle cell anemia, for example. Effects of anemia, we see tissue hypoxia. This basically means the tissues aren't getting oxygen. That could, but not always, lead to necrosis, which is tissue death from lack of oxygen. Patient's going to have shortness of breath and be lethargic if their tissues are hypoxic, can't function very well. Also, we see low blood osmolarity and low blood viscosity as well with anemia. Now, sickle cell disease, again, is one of those hemolytic anemias. It's a condition where that's inherited. So you can only get sickle cell anemia if both parents are carriers of it. It's a recessive allele for you geneticists in the room where there's a 1 base substitution in the code for making hemoglobin, and therefore that causes the shape or the structure of the hemoglobin to change. A person who has sickle cell disease has usually a shortened lifespan, although things have gotten better. The sickle cell hemoglobin, of which they abbreviate HbS, turns into this gel-like substance in low oxygen concentration. So if you have a high oxygen man, so that you're emptying the oxygen off those red blood cells, that makes that hemoglobin get gel-shaped like, and it can cause the sickle cell shape to happen in the red blood cell. This can result in agglutinating or clumping of those cells and blocking vessels. That means now you could be intense pain, heart failure, paralysis, anemia, hypoxemia, all can occur during those episodes. Now, a person who has sickle cell disease typically does have to have both genes, one from a mom and one from a dad for sickle cell. People with only one gene for sickle cell are called carriers, but they actually have some sickle cell hemoglobin in them, just not enough to have or present any symptoms of sickle cell disease. Those individuals have an advantage over malaria because malaria doesn't like or can't ingest sickle cell hemoglobin. So malaria, which is a parasite that lives inside of your blood cells, will not infect or will not be able to live or grow inside a person who's a carrier or a person who has sickle cell disease. So those guys have a benefit against malaria. Okay, if you have sickle cell disease or you're a carrier of sickle cell, no malaria. But of course, if you have sickle cell disease, you're going to die from malaria, die from sickle cell, so that wouldn't be very good either. Now, thalassemia is another abnormal hemoglobin. This is also hereditary defect. It is more common among people of Mediterranean ancestry, and this is one of those or results from one of the globin chains to be absent or faulty. That results in the red blood cells being really thin and pale and very delicate because of the deficiency in the hemoglobin. And you can see here how the center of these red blood cells are almost look like they're hollow, but it's just real thin hemoglobin and makes them look more transparent. Now the opposite of a low count or low red blood cell count, which we see with anemia, we have polycythemia, which means we have excess of erythrocytes. You can see them all packed in here, lots, way too many. If I have a huge number of red blood cells, that's going to make my blood very viscous or thick, and so it doesn't move very well through the blood or through the blood vessels. Now there are different kinds of polycythemia. One of them is polycythemia vera. This is a bone marrow cancer that results in excessive production of blood cells, and so your hematocrit may go up as high as 80%. Remember, the norm is 45%. So that would be very viscous blood. That can cause blood clotting because the viscous blood can't move very quickly. The other type of polycythemia is secondary polycythemia. This is just an adaptation to living at high altitudes. So again, if you move to Denver, you're going to end up producing more red blood cells in response to the low oxygen levels in the atmosphere. And so you end up having higher red blood cell counts. You can also get a secondary polycythemia basically by having excessive EPO production. Again, this is not necessarily a normal thing, though. This could be an abnormality in the kidneys, or it could be high excessive EPO production in response to low oxygen living at high altitudes again. And then that again ties in with blood dope, where athletes take it as a performance enhancing drug. And the problem of taking too much EPO to enhance your performance means you're going to end up with polycythemia, or high excessive number of red blood cells, which means you have very viscous blood, and that could lead to strokes or heart failure. So that's going to end our video on erythrocytes. The next one we're going to do then is going to be on white blood cells and platelets.